Good evening, everybody, and welcome to BrisScience, hosted by the University of Queensland and here at the fantastic Edge at the State Library of Queensland. I am your MC for this evening, Joel Gilmore. Thank you for coming, Mum. I'd like to start by respectfully acknowledging the traditional owners of the land on which we're meeting tonight and pay our respects to elders both past and present. Tonight, of course, we have a fantastic speaker who we're going to introduce in a moment, but a couple of bits of housekeeping. If you have a smartphone, set it to silent, but get ready to ask questions using hashtag BrisScience. We will be live tweeting, I should say, all throughout the evening, so you can have a record of some of the things later. And of course, this will be on YouTube later on. If you don't have a smartphone, that's okay. You can also ask questions at the end through paper. So with some question slips on your way in, or if you have a signed photo or something you'd like to submit, feel free to write your question on that, and we'll come round after the talk and collect those, get through as many as we can. And finally, there is food and drink afterwards, and we'd love for you to hang around it and have a chat amongst yourselves, but also to our speaker for any final burning questions. I think that's all the housekeeping. We'll talk more about next month's talk after this. So tonight, our talk is Pathogens, Pandemics and Preparing for Disease X, with the slightly terrifying subtitle, The Next Pandemic is Inevitable. To help us understand more, I'm very excited to introduce Professor Paul Young from the University of Queensland, where he is Professor of Virology and Head of School of Chemistry and Molecular Biosciences. He and his group have more than 40 years' experience studying diseases, and so is ideally placed to tell us what might come next and how scientists are responding. And of course, as I said, this is going on YouTube, so this could be UQ's next big viral video. Thank you. And if so, we are in exceptionally good hands. Please put your hands together for Professor Paul Young.
Thank you, Joel, and I promise no more of those. It's a fantastic privilege to be here to present some of the work that we're doing in our laboratory in preparing for Disease X, but also to bring some of the excitement of the research that's going on at the University of Queensland. So what I want to do today, before I start actually, I always like to start with this slide. This is my group. An amazing group of talented individuals. All the past and current members of the young group essentially combine the talent and output of the group. I want to particularly mention three people here, Keith Chappell, Dan Mortensen and David Muller, who are senior postdocs in the lab and really are doing and driving all of the work that goes on in the lab. What do? We understand how viruses cause disease. That's at the core of our research endeavours, to try and understand how viruses interact with their hosts at the cellular level and also at the organismal level. But we're also, in a translational way, developing strategies to control virus infections that cause disease. And I'm going to come back to that particular point a little later, because I want to put you all in the context, provide some context for viruses and their engagement with hosts that isn't just all about disease. It's pretty much what most people hear about viruses, but actually there's a wider community of viruses out there engaging in their hosts that aren't actually causing disease. And I want to just raise that as a starter for the conversation about the pathogens part. So we're looking at ways of controlling them, and that's in the areas of diagnostics. We develop diagnostics, we commercialised and have out in the field an early detection for dengue that has been used now for the last 10, 15 years as the global lead diagnostic. But we're also working on therapeutics, drugs that can treat actively infected individuals, but also vaccines that can offer prophylactic or protection on subsequent exposure to diseases. So that's what we do as a group. Amazingly talented group of people. The way I want to break this talk up today is into three components. We'll definitely talk about the pathogens and the pandemics and preparing for disease X. But I wanted to start off by placing viruses more in context for you. The viruses you don't tend to hear about much, the good viruses, the ones that actually are engaged with us all the time, that actually have some beneficial effects for us. All hosts, in fact. There are viruses of plants that actually assist them in their engagement with their environment. There are definitely the bad guys, though, and the really ugly ones, and they're the ones that potentially can cause these diseases and ultimately potentially a pandemic. So we'll then talk about those, the emerging viruses, how they actually come about, why are they not good viruses, and then I'll finish off with a little bit about how the world has changed its view on how we deal with epidemics and pandemics or emerging outbreaks. And this has really only happened in the last few years. There's been a concerted effort to really start thinking about stopping the reactionary approach we have. Oh, it's never going to happen. Oh, it's happened again. What do we do about it? To actually starting to prepare for it. And I'll talk to you about some of the approaches we're taking. So this is just a global map showing some of the viral, bacterial and parasitic pathogens that have emerged over the last 20 or 30 years. As you can see, it's spread right across the globe. Whoops, I knew I'd do that. You'll notice this one here, our own Hendra virus, which was isolated from horses and infected individuals, humans who came into contact with those horses in the suburb of Hendra. That's where it was first observed. We've only seen a handful of cases since, but a relative of Hendra emerged in Malaysia called Nipah, which has gone on to cause much more serious and wider epidemics and pandemics. It's spread to India, and it's also now spread to the east coast of Africa, carried by its host, the bat, following its migration routes. So these viruses have emerged over the last 20 or 30 years and cause local epidemics and in some cases pandemics where you get spread across the globe. And you can see I've colour-coded them here. Red represents newly emerging viruses, ones we've never seen before. Blue are the re-emerging viruses, so we've seen them before, maybe in small little outbreaks, maybe 1 or 2 cases of human disease, and then all of a sudden they expand. We knew about Zika virus back in the 1940s. It was first isolated in Africa in the 1940s. Really didn't give it much thought. It was just catalogued away as just another virus that's been isolated over time. But then there was an explosive outbreak in 2015-16 in South America. Coming to grips with how that happens is part of what we do. There's also one here of deliberately emerging diseases, and this relates to bioterrorism acts. There are a number of viral pathogens in particular that are on lists that are potential bioweapon agents, but the only time we've ever seen this in reality is the release of anthrax. I thought I'd start off with these quotes. One is from Sir Macfarlane Burnet, who was one of Australia's Nobel laureates. He discovered some of the basic aspects of our immune response and is highly regarded for all of the work he conducted. However, he did say in 1962 that one can think of the mid-20th century as marking the virtual elimination of infectious disease as a significant factor in life. Another quote from the same decade was from the US Surgeon General, who said it was time to close the book on the problem of infectious diseases. Not great foresight. But you can understand why they were thinking that in the 1960s. The 1960s was that period. We've had 15 to 20 years of antibiotics. Antibiotics really only came out after the end of World War II. And we were developing new antibiotics all the time. The idea of antimicrobial drug resistance, antibiotic resistance, hadn't really come onto the scene. So new ones were coming along, and we were combating all of these age-old bacterial diseases and eradicating them as problems in the communities. So that was one side. The 1950s and 60s also saw the emergence of quite a number of vaccines, particularly childhood vaccines. So we were seeing the eradication of things like polio, close to eradication of polio, certainly the removal of polio from many communities as a significant disease. Those old enough to remember the iron lung wards in the Royal Brisbane Hospital will know that prior to the vaccine, polio was one of those scourges that was amazingly feared. Measles was sorted out by a vaccine. It's coming back again. That's a whole other topic, which I won't get into tonight. But vaccines and antibiotics were probably what were leading them into this frame of mind. However, what we have seen is, in fact, increasing outbreaks. Over the last 30 years, we've seen the number of infectious disease outbreaks worldwide rise steadily. Mostly this is because of viruses and bacteria. Such that now, if you look globally at deaths, there's about close to 60 million deaths a year. If you actually look at what all the causes are, the major cause of disease is cardiovascular diseases. I'm just thinking how focused that is. It's cardiovascular disease, but not far behind it as the second leading cause of death globally are infectious diseases. Now, many of the figures that go to make up this particular pie chart, and particularly in the infectious disease category, are in the developing world. There's far less infectious disease in our communities. However, there's no reason for complacency, because if I showed you a graph, which I don't have up here today, but if I showed you a graph of infectious disease in Australia, it is also going up. There are a number of reasons for that, not least of which is antimicrobial resistance, or the development of it, and the fact that we're not developing new antimicrobials. So what I wanted to do first, though, and I alluded to this right at the start, I want to give you a bit of a background on viruses, just to place into context what pathogen, what is meant by the word pathogen, and the causation of disease once a virus infects its host. I think most people, what we hear about viruses is that that's the normal course of events. It's not. Viruses come in many shapes and sizes, somewhat beautiful up here, rendered in different colours. We can't actually tell the colour of these viruses, but that's showing the different proteins that make up the outer coat of these viruses. These are called, these particular ones are called naked viruses because they don't have a membrane around them. Some viruses have a membrane. They can be filamentous, like Ebola, or rod-shaped, like this virus that infects tobacco leaves. In fact, tobacco mosaic virus was really the first virus identified as an infectious agent back in the late 1800s. They can have an envelope, and what you see here is a string of human immunodeficiency virions, HIV, budding off the surface of the cell that they'd infected. And you can see that they're taking with them an envelope as they bud and mature and spread from one cell to the next. Or they can be incredibly complex. They can actually look like lunar modules. And this is a real structure. There are viruses that actually do look like this. It's even more bizarre. This sheath on the outside here is made up of molecules that can change their shape when given energy. And the column twists and it reduces its size by a half. So what happens is that this little central chamber here gets punched through the membrane of the bacteria that it's infecting. So it's like a syringe. It literally punches a hole into the cell and injects its nucleic acid. So there's a lot of variation. But what they do have in common is something very simple, and that is that they have a genome. That's their information. They encode genetic information. And they're wrapped up in a protective coat. And that provides protection in the outside environment, outside a cell, where there are nucleases that could easily chop up the genome. That's it. That's pretty much a virus. Genome, protective coat. In terms of their replication, forget about all that bit. All that was meant to show was you've got one of these virus particles, it gains entry into the cell, it replicates, and produces progeny, which are released. So really the life of a virus is about these virion particles that just go from cell to cell. So essentially they're just mobile genetic elements moving from cell to cell. In fact, it provides one of the debates that virologists over a beer will debate, and that is whether viruses are actually alive or not. Because in this state, they're essentially just inert macromolecules. They're just bunches of protein with a piece of nucleic acid inside. They don't have any motility at all. They literally get from cell to cell by random movement, so the environment will take them there. It's only when they get inside a cell that that information is translated and there's progeny. I mean, my argument has always been they evolve, they replicate, so they must be alive. I've come to a nice balanced response. I think they're just like Schrodinger's cat. I think they're both dead and alive. They're dead out here and they're alive in there. Viruses infect literally all living things on this planet. What might surprise you is for some of the really big viruses, they have viruses that infect them. So we even have viruses of viruses. We eat and breathe billions of viruses every day in the food and drink we ingest and the air we breathe. I'm sure you've heard, there's been much news in the press, and I know one of these talks by Phil Hugenholtz told you about the gut microbiome. So we know our microbiome, which we now know is incredibly important to our own physiology, are controlled by viruses. There are many more viruses than there are gut microbes in our gut, and they are there controlling that particular population. You may or may not know, and might be surprised to hear, that there are viral genomes actually embedded within our genome. All of us sitting here carry a number of viruses that are permanently embedded in our genome that we pass on to our offspring. And I'm going to spend a tiny bit of time on that just to explain another angle on this good viruses story. As I said, not all are bad. In other words, the bad meaning causing disease. You might not know that there are between 1 and 10 million virus particles in every milliliter of seawater. I've just been down the Gold Coast over the weekend, and it was nice, crystal clear water. Maybe not on Saturday when it was raining and 14 degrees, but yesterday it was beautiful in terms of clarity. We would be living with a sea that was a huge soup of bacteria and protists if it wasn't for the viruses keeping them under control. So there's a huge number of viruses, and you can see some of them here that have been taken from marine sediment. There's something like, estimated to be about 1 million different virus species, not just 1 million viruses, 1 million different virus species in 1 kilogram of marine sediment. We didn't know about this till about 20 or so years ago when someone actually went out into the sea and actually took a couple of litres of water, filtered it down and then analysed it, discovered all of these new viruses. You may also not know, you won't be able to read any of that, but I'll just briefly summarise it, that viruses actually contribute to the global carbon cycle. Depending on the level of lytic attack of viruses on protists and also bacteria that are in the ocean, will define whether carbon sinks to the ocean floor as particulate organic matter, or whether it stays in the upper reaches of the sea as dissolved organic matter. And that dissolved organic matter ultimately finds its way through carbon dioxide release into the environment. It then gets sucked back into the ocean as well. So there is this cycle of carbon dioxide that is somewhat controlled by viruses. I'm pretty sure I once had the opportunity of sitting next to a climate modeller at a dinner in Canberra, and I said, "Do you actually have viruses in your models?" And he just looked at me. So I suspect that's still the case, that they're not actually modelled in. We can get giant viruses. These were only discovered about 10, 15 years ago. For size comparison, here is a little virus called rhinovirus, which causes the common cold. When you get a common cold, most of them are caused by members of this family. HIV is here, and here are these new viruses. All of these new viruses, there are 3 families that have been identified so far, all infect amoeba. So they haven't been found in us yet. But the amazing thing about these is they totally rewrote the books on virology in terms of size of the genome. We always thought that viral genomes were much smaller than bacteria, so we had animals and plants, and then down to parasites and bacteria, and then viruses in terms of size. But these things have genomes that are in the megabase size, And some of these viruses have genomes that are bigger than some bacteria. So we've now got an overlap in these two kingdoms. There's a proposal put forward that these viruses, the mimiviruses, are actually a separate part of the tree of life. One was isolated about 5 or 6 years ago from a Siberian permafrost. And the way these studies are done these days is various unusual environments are searched for exotic pathogens. And they usually do it by sequencing. So you just take the fluid or the tissue or the sample, extract all of the nucleic acid, sequence it all blindly, and then through the power of bioinformatics, AI and algorithms, we can get a picture of the genetic population of organisms within that particular environment. So that was really exciting. They found a new giant virus in this permafrost, which was carbon dated to 30,000 years. And then they did the unusual experiment. I'm not even sure I would have tried this, but they looked to see whether they could grow it. And they added it to some modern amoeba, and the thing grew. So this is a 30,000-year-old virus taken out of permafrost that is now replicating. And it led them to say, in the last paragraph of their paper, that the thawing of permafrost, either from global warming or industrial exploitation of circumpolar regions, might not be exempt from future threats to human or animal health. The suggestion that there might be ancient viruses in there that might have the potential to spill over into animal or human populations. I think that's a long bow to draw. Certainly the virus that they pulled out will only infect amoeba, so I think we're safe for the time being. But I think you get the impression, or the story here, that viruses are amazingly resilient. So you've heard about the human microbiome. It's essentially the aggregate of all microbiota that reside on or within human tissues and biofluids. And that's not just the gut, which is the one you've probably mostly heard about. There are bacteria all the way through us. And we even now know that there are bacteria circulating in our blood. That used to be a sign of disease, but in fact there are commensal bacteria that are circulating in the blood. But if you look at and if you compare the number of genes encoded by that microbiome, it's about 100 to 1 over our own genetic composition. So this microbiome is made up of trillions of bacteria, fungi and other microbes, and as I said before, it plays an important role in health through modulation of things like metabolism, inflammatory responses, the nervous system, and as each day goes by and more studies are done, we're understanding more and more of the impact that these bacteria have. But remember, all of them are in turn controlled by an even bigger population of viruses, and we're really only starting to scratch the surface in terms of understanding the implications that has in terms of how that control manifests itself. So in terms of the viruses, we're now starting, and this really is early days, we're now starting to see or envision the concept of a human virome, or any environmental virome, quite frankly, for that matter. Probably the really surprising study came, it was about 6 or 7 years ago now, with this amazing new capacity we have for what we refer to as deep sequencing. So this is where you can just take an environmental sample or a clinical sample and literally extract all nucleic acid and sequence it blind. We hadn't had that technology up until relatively recent past. Prior to that, we would go into individual samples and target them with specific sequences of pathogens or infectious agents we knew, and we'd pick out, oh yes, that's there or it's not there. This is done blind, so total sequencing, and then you run it through algorithms that throws out, yes, you've got this, and this. Oh, and this is new. It looks like this, but it's still new. So ways of identifying what we've got. This is what was found when someone looked at human blood and a number of different tissues. You don't need to see the names. I just wanted to show you the numbers. So this is the digestive tract, the respiratory tract, and here is blood. And each and every one of these is a virus family for which representatives were identified in a wide range of individuals. And just picking one of those out, just as an example, it was a virus that was discovered by doing this. We didn't know about it before. It's called Anillo virus. We now know that every single person in this room has it circulating in your blood right now. It's a commensal virus. It's never been affiliated with any disease. We don't know that it causes disease. If anything, the latest research would suggest that it does a little bit of tickling of our innate immune response, a little bit of stimulation. So we're actually better prepared when we actually meet an infectious pathogen, when we meet a pathogen or a virus that has potentially deleterious effects on us. So no disease association, but a function yet still unknown, and yet it's in every one of us. So we're starting to discover that these viruses are everywhere. The last example I'm going to give is probably the one that's going to amaze you most if you haven't heard this story before. And this came out from the Human Genome Sequencing Project, which ran for about 12 or 13 years through the 90s. And didn't finish, it sort of started a whole new era of research in 2000 when the first draft human genome was published. Since then, more genomes have been published. That cost a couple of $2 billion to do over 12 years. You can now sequence a human genome for less than $1,000 in a day. So technology has moved dramatically now. But what was discovered there changed our view on life. There were two major things, from my perspective, that came out of that sequencing analysis. The first one is that only 1.5% of our entire genome actually codes for proteins, which we used to think were the major parts of the information message that DNA carried that made us who we are. 1.5%. There has been terminology thrown around for some time that a lot of DNA in our genome is junk. But because it doesn't code for protein and we don't know what it does. But the more we research this, the more we're finding that all of this DNA actually is probably important and does other things, but it doesn't encode protein. The other quite remarkable finding was this one, that 8%, nearly 10% of our genome is actually the fossil record of ancient retroviruses that invaded our ancestors or infected our ancestors, that became embedded in our genome, that then been carried down through the generations. Some of them are full remnants, the full genome copies, so you can almost see the entire virus within our genome. But many are just remnants. They're now just passengers in our DNA. They're just the record, the fossil record, as I said, the molecular archaeological record of a past, very ancient, sometimes many millions of years ago, infection event of our ancestors. To understand this a bit, I'm just going to take you, this isn't a virology lecture, so I won't be taking you through any detail. This is a retrovirus life cycle. These embedded genetic elements are mostly retroviruses, and it's because of their particular life cycle when they're normally replicating. And so I only want to pick up 2 points. The first is to deliver their genome into host cells. The virus, these envelope viruses, these are the sort of viruses that have an envelope, have in their surface a very efficient machine, an efficient fusion machine called ENV. It's actually short for envelope. So it's just a protein embedded in the viral envelope. I'm going to come back to that in a little while, so that's why I'm emphasising it. But it's the protein that drives the fusion between the viral membrane and the host cell membrane to allow the virus to infect, because it's got its genome inside this coat. It's got to get the genome from out of the coat into the cell. So what it does is it fuses. So you then get infection, and through various pathways that I won't go into, eventually the viral genome gets inside the nucleus of the cell. And what it does then is it inserts its genome. It's got some little enzymes that chew the host chromosome and insert its own DNA so that when the host chromosome gets replicated, the virus gets replicated as well. And that's part of its life cycle. RNA transcripts come off, the viral genome gets packaged, and more viruses flow out. And you can see those up here. So that's the full virus life cycle. But this is the key point. If the retrovirus can infect germline cells, for example, egg cells or sperm, remember I said they're permanently in there. Once the virus inserts its genome, it doesn't come out again. It's permanently there. I mean, for most cells, that doesn't really matter. HIV is like that. It doesn't infect germ cells, thank God. HIV will actually integrate itself into the host chromosome, and you'll be carrying that for life. It's one of the challenges with developing strategies for combating HIV. You're always carrying it, unless you can completely get rid of all of the cells that carry that genetic message. But if it's in eggs or sperm, and because it's permanently integrated, you're obviously going to pass that on to your offspring. So that will then happen for all subsequent offsprings. So the 8% of the genome that's retroviral is simply the remnants of those past infections of our ancestors. So if you actually look at our cousins, the primates, we share some of those elements depending on when we split off the family tree. There are examples of viruses that have entered into our host chromosome after we split off from our primate ancestors, and so you'll only find them in humans, and all of us will carry these HERV-Ks. Won't worry about the nomenclature, but it just means human endogenous retrovirus. But these ones that inserted themselves into our ancestral DNA 20, 25, 55 or 100 million years ago are obviously shared by our common ancestors on this family tree. So this HERV, for example, is found on the exact equivalent chromosome in the exact equivalent places as it is in these other primates. So it's a bit of molecular archaeology. You can actually trace when the different primates split off from each other by whether they've got some of these envelope sequences. So over time, we're talking millions of years here, these are no longer viruses that are replicating, they're simply embedded genetic elements. They can acquire mutations and deletions so that they're no longer functional. And so they're merely genomic passengers. However, we do have some examples where some of them can be deleterious. Some cancers have been linked to the upregulated activity of some of these endogenized elements. But they can also be beneficial. And this is the kicker. This next story, if you haven't heard this, will blow you away. It's this retrovirus. It's called HERV-W, similar nomenclature as all the others. And this is what the genome would have looked like when it inserted itself. These little wiggly lines are the human chromosome, and the virus has inserted itself here. Won't worry too much about these nomenclature. These simply mean they're names for different genes. But you'll notice the N one, right? So I talked to you about the N before. The M gene encodes the protein that drives the fusion. It's a fusion machine. This is what it looks like now in the human chromosome. GAG and POL have acquired deletions and mutations that mean they're no longer functional. So this cannot come out as a virus. It can't emerge out of the chromosome as a virus anymore. However, what was discovered, and this was discovered in that first edition of the human genome back in 2000. We didn't know what it did in those days, but we've now subsequently worked it out. It's a full-length open reading frame for a protein. It's got a start codon, a finish codon, so it's fully intact. Essentially the envelope protein of a retrovirus. When they looked at all the different tissues as to where it's expressed, it was found that it was expressed in highest levels, in fact almost uniquely, in the placental tissue. The gene is now called syncytin. It causes membrane fusion. What does it do? When an embryo implants into the uterine wall, it ultimately develops an umbilical cord. How that actually begins is the development of a single layer of cells called the syncytiotrophoblasts. And that is a fused cell population. So it's the one cell, multinucleated, that's been fused. The protein that does that is this viral envelope protein that was once a virus. So we are placental mammals because of this virus. This is what it looks like in the very early stages of a blastocyst entering into the uterine wall. And that syncytiotrophoblast is absolutely critical because what it does is it provides the exchange of nutrients and waste for the embryo. One other additional fact, I'm probably running out of time to talk too much about this, but it's so cool. These envelope viruses also have embedded within them sequences that suppress the immune response. It's one of the reasons why retroviruses can be immunosuppressive and cause disease. But one of the issues that has always intrigued those working in reproductive biology is embryos are often not tissue matched with their mother. Why aren't they rejected? It turns out that this envelope protein also contributes in that way by local immune suppression to allow it to engage and fully develop. So the main reason I'm here, let's get on to that. But the reason for actually raising all this, of trying to give you a better impression of this virus-host interaction is not all about causing disease, is that evolution tends to drive stasis between these agents. What is driving the evolution of both the host and the virus is some sort of reproductive advantage. You don't get a reproductive advantage by killing your host, okay? So what we find is that most virus-host interactions that have been going on for a long period of time, both the host and the virus have evolved to low pathogenicity, low disease. They become commensal. In some cases, as you saw in the previous example, they've actually assisted with our own evolution, so they can be highly beneficial. So natural selection favours viruses losing that disease-causing capacity. It's quite possible that when retroviruses first engaged with us all those millions of years ago, it wasn't good for us. We didn't know that at the time. We did know that because it was an ancient event. I'm involved in a research project. We discovered that there was an endogenous retrovirus in koalas. And through various studies, what we found was that it's currently invading the koala genome. And it's spreading through the koala population from north to south. Up here in Queensland, every koala has this virus integration, but down south they don't. And certainly we're seeing disease. We're seeing high rates of cancers, high rates of opportunistic infections like chlamydia, but there are also populations of koalas that have the virus that are very healthy. So they're already starting to acclimatise to having this virus with them. But it's not been smooth sailing. They're actually suffering from this virus at the moment. So they don't eradicate their natural hosts. What's the point of that? Remember, it's a mobile genetic element. From an evolutionary perspective, what it wants is a host that's able to transmit efficiently. I suspect if a virus that's embedded itself into the genome of its host has reached nirvana, because it's basically there forever and it just will continually pass on. So by and large, disease is a somewhat unusual consequence of infection, not the common outcome. So where do all these emerging outbreaks come from? It centres around the fact that these virus-host co-evolutionary interactions happen between single virus populations and a single host. And that is an evolutionary process that's happened over a long period of time. What happens if that virus accidentally spills over in a completely different host it's never really encountered before? The consequences can be dire because they haven't actually reached an equilibrium yet. And that's how most of these happen. You have viruses that are embedded usually in wild populations. Here we've got represented monkeys, bats, and rodents. And each of those are perfect examples of viruses that have spilt over into humans. Ebola is a bat virus, as is Hendra. Hendra in bats doesn't cause overt disease. Most bats can carry it without any problem at all. But when it gets into humans and horses, it's a significant problem. It can be aided by perhaps transfer into domestic animals first, and the spillover can happen from domestic animals into humans, and it can be mediated by vectors as well. So those viruses that are transmitted by mosquitoes or ticks can help in the transmission across this cycle. So in terms of a time event, you can have viruses meandering along in the wild animal scenarios, and then you can get these spillover events into domestic animals or directly into the human population. Or it can come via domestic animals as a spillover event. In each one of these, you get local amplification. The problem we have had with emerging outbreaks is this is the time we usually are aware that something's amiss. We don't really recognise the spillover event until it's well established in the human population. We've got significant numbers where it's exploding in that population, and we just don't have the time to prepare for it, or time to combat it. So how do we prepare for it? It certainly generated a lot of headlines, and Disease X, WHO's naming of an unknown pathogen, has really helped that. It's not Z, it's Disease X. They don't cause zombies, except if you go and see certain movies. But there is concern, obviously, and rightfully, as to the consequences of these emerging outbreaks and what they can do. So the last 20, 30 years has seen sudden and dramatic emergence of a wide range of these previously unknown or known viruses into new geographic locations and or in greater numbers. Things like Ebola, for example, we've known since the 1970s, but it's been a trickling outbreak. It spills over from the bat and the local primate population, usually into villages. What was a big surprise was in 2014 when it exploded out into a larger city environment, and that's when it really caught hold. And we haven't really curtailed it yet. It's been up and down, and there's a new outbreak of Ebola happening right as we speak. But there are a number of examples here. West Nile virus was unknown in the Americas until 1999 when in New York, crows started falling out of the sky. They believe from sequencing that it came from the Middle East, but they don't know, from Israel in fact, but they don't know how it got there. Some mosquitoes hitching a ride on a plane was thought to be the most logical way that it was introduced into the Americas. It then swept across the Americas, killing thousands of individuals and infecting tens of thousands. And it really hasn't gone away yet. Chikungunya is a virus a bit like Ross River that causes joint pain, and that spread globally in the 2000s. Middle East respiratory syndrome appeared in 2012 in Saudi Arabia. We didn't know this virus before it spilled over from the camel population into camel herders. But there's been ongoing outbreaks ever since human-human transmission, including an unexpected outbreak in South Korea. Ebola, as I said, has been drifting along for some time, as has Zika, which emerged suddenly in South America, and now yellow fever's coming back. Just as we have a yellow fever vaccine shortage, it's coming back in Africa and also back into South America. What are the things that have changed? It's a multitude of things, which is what this figure on the right-hand side is meant to indicate. Physical and environmental factors, the genetic factors of both host and the virus, human and animal issues, close juxtaposition, for example, to where the pathogens may emerge, and so on. So changes in our physical and biological environment, climate change is definitely impacting, particularly vector-borne diseases and the amplification, for example, of the mosquito populations. Diet and habitats, deforestation is taking humans into close proximity to some forest environments which may harbour these exotic pathogens. We know of pathogens that have emerged that way. In South America, the hemorrhagic fever virus, Hunin, is a virus that had reached its coevolutionary relationship with rodents that lived in the forests. And when they started clearing the forests and started planting wheat, the rodents came out into the wheat fields and engaged with the farmers, the wheat farmers. And suddenly we started seeing this hemorrhagic fever virus appearing in those farmers. So our own demographic and behavioural changes have affected disease transmission. There's been an unprecedented population growth, and of course, modern transportation has really helped to transmit viruses around the world. And of course, evolution of viruses and hosts themselves, making them more susceptible and allowing infection where previously it may not have happened to occur. This gives you an idea of the potential for spread of a viral pathogen in a human vector. So you pick up a virus infection somewhere in the world, within 48 hours you can be anywhere. And that's the incubation period of many viruses. So you can be incubating it, not showing that you have that disease, and go to another country. That's exactly what happened with SARS. It came out of the civet population, We now know it was actually bats that infected civets, and civets in Hong Kong infected a large number of individuals, some of whom flew to various parts of the world and initiated outbreaks in those areas. Toronto had a large SARS outbreak as a result of one plane travelling to Toronto from Hong Kong. So this provides the perfect potential for spread. I took this, you might want to visit it yourself. It's called Health Map, and it shows exactly what's happening in the world as you speak. I did this last night. So these are the current outbreaks that are occurring around the globe as we speak. How do we get this information? There's a wide range of bodies that are accumulating this information and collating it all. You have, I visited all of these, and they're a bit like going into a movie set where they're run 24/7, they've got big screens around the side, and they're connected to all of their field operatives who are giving information back. They're also more and more utilising things like mobile coverage. So we're getting a lot of information out of disease spread just by recording mobile messaging. But you have in Geneva at WHO, in the US and the China CDCs, the same things going on, monitoring, and then people going out to look at curtailing those outbreaks as quickly as possible. So it was in 2016, a couple of years after the Ebola outbreak emerged in Africa, the WHO was yet again responding to criticism that all they were doing was being reactionary to disease outbreaks. It was a fair cop, and they accepted that. But they initiated a working group to work out, well, what could they do? What could they do better? And they came up with the idea of a preparedness strategy. And one way in which they could do this is to actually develop a list of pathogens they knew that were ticking away, little ticking time bombs in Africa and other areas of the world that had the potential for emerging. And so you see they came up with this list of 9 diseases. There are 2 here, MERS and SARS. And for the first time, someone actually recognised this potential for an unknown pathogen to emerge. So disease X simply represents the potential that an unknown pathogen may be the cause of serious epidemics in the future. We know it's going to happen because it has happened in the past. That's exactly what MERS is, it's exactly what SARS is, exactly what Nipah is. We've never heard of these viruses before. We only found them when they suddenly emerged into human populations causing disease. WHO is more a strategic policy-driven process than anything else. They don't have the funding to actually drive this analysis. So onto the scene came CEPI, the Coalition for Epidemic Preparedness Innovations. Not the best name in the world. Let's just call it CEPI for now. But this is a group made up of the Wellcome Trust, the Bill and Melinda Gates Foundation, operating through the World Economic Forum, and also funded by a number of funder countries, including Australia, but the big funders are Norway and Japan and Germany. They've now raised or accumulated something like $800 million. Their goal is to get $1.5 billion that they can put into strategies for preparedness. And that preparedness looks like this. They have 2 main priorities. We'll develop vaccines now rather than wait to see which one will come out. They'll develop vaccines to a range of potential emerging known viruses, and these are the ones they've got on their list so far. They will add to them. It's only been running for 2 years, so they're now working on that. And there are multiple groups working on this. They've provided funding to biopharma and academic groups to actually develop this. They've also taken the approach of looking at and funding platform technologies that could be used and rapidly deployed for things like disease X. So it's more, the vehicles for delivery of new knowledge as it emerges when an outbreak comes about. Just want to highlight again some of the team players. Keith Chappell in the group, one of the senior postdocs in the group I was mentioning, is really driving our program, CEPI program, along with Dan Mortensen. And this is our consortium group, which includes people from CSIRO, University of Melbourne, Australian National University, University of Hong Kong, and a number of others, where what we're trying to do is develop a pipeline, a platform pipeline, which is a rapid response vaccine preparation. Now, I'll just briefly finish off by telling you how we're doing this. Viral surface proteins are the main target for antibody responses, the adaptive immune response. Okay, so not surprisingly, they're on the outside, antibodies will bind to them. For envelope viruses, they carry out membrane fusion to facilitate entry. Right? So getting back to that envelope protein that drives this fusion process. It's also the target of the most potent neutralizing antibodies that will kill it if you've got a good immune response. The problem is that these proteins, to drive the fusion, they are literally machines that flip through into two quite dramatically different structures. And all previous attempts at making vaccines based on just expressing or taking this little piece, rather than the whole virus, you just take the outside protein. Whenever you do that, they naturally fold into this structure that's the post-fusion form. Trouble is, that's not what's on the surface of the virus, so the immunity you induce to that doesn't actually protect very well. So the challenge for quite some time in subunit vaccine development has been, how do you actually lock that structure in this pre-fusion form in an easy way that can lead to a vaccine? Let me just take you through some cartoons that shows the principle of this. When a virus infects us, our immune system responds, okay? Generating antibodies and cellular responses that clear the infection. So you get antibodies, you also get cells that will target infected cells and clear them. It also establishes memory. So once you've seen a pathogen, you retain cells that have a memory, such that next time you see that pathogen, they will quickly come into play, they get amplified very quickly within 24 hours and can protect you. That's why, when you get one infection and you develop an immune response, you tend not to get it again. There are some exceptions to that, but that's how a vaccine works as well. If you acquire an infection naturally, you tend not to get it again because you've got memory. The idea of a vaccine is to do that. The most potent antibodies, as I've just said, recognize multiple sites on these virus surface proteins, and they do what we call neutralize it. So the best vaccines elicit an immune response that closely mimics what you would see from a natural infection. That's the whole basis of vaccination. We try and induce an immune response that's the same as if you've got the disease, but without the disease bit. Okay? And that's how all vaccines work. One way to make a vaccine, as I've already mentioned, is to simply use those surface proteins. Unfortunately, these ones are the unstable ones. They fall apart or change shape, so they don't induce the right response, and you don't get effective neutralization. The current influenza vaccine is exactly that. It's good in that it's better than nothing, but sometimes it doesn't induce strong enough immunity. One of the vaccines we're developing is a better influenza vaccine that will lift that efficiency. So what we've done is develop a molecular clamp. This was an idea that Keith Chappell in the lab came up with, and we've now developed it to a point that we've been able to apply it to a number of different viruses. It locks those recombinant proteins into their appropriate form, and I've just sort of schematically represented it as a little clamp down the bottom of the protein there. That teaches the immune system to induce antibodies of the right sort that will protect the virus. So we've now shown that this works for a wide array of viruses, and so that can provide the basis for a generic platform for a wide range of known viruses, but also unknown viruses. We just plug it into the system. So it could include those to disease X. And you can see down here a picture of the real proteins and how we can clamp them. The red bit is the clamp. Not that this is detail you need to take in, but this just shows you we made vaccines to a wide range of these viruses now. Here's an influenza one, respiratory syncytial virus, which is a major pathogen of young infants, the MERS, Nipah, a herpes virus equivalent, and also Ebola. And the fact that they're nice and separated and homogeneous means that they're likely to be a good vaccine. Now the problem. Vaccines normally take years, even decades, to develop and deploy. What CEPI proposed in their application, they said, only submit an application to us if you can target a 16-week time frame. You identify the pathogen, the antigen, to a product release for clinical trials in 16 weeks. Completely unheard of. Also, that we would have the capacity to produce at least 100,000 vaccine doses that we could implement. The whole idea of a rapid response vaccine approach to an emerging outbreak is a thing called ring vaccination. So you don't want to vaccinate the entire population, so you don't need the millions of doses. But where an epidemic is happening, if you can generate a vaccine quickly enough, you ring vaccinate around the outbreak response so that it stops the spread. So again, I'm not going to go through the details of this. This is our pipeline of activity. So from pathogen identification, we'll make the antigen within 4 weeks and then test it within 16 to a point that we can take it into humans. And that's challenging, but we've shown that with a number of demonstrator viruses we can do it. So our task over the next 2.5 years, and Keith, as I said, the project co-lead on this, is to validate this with 2 human clinical trials, and we're going with influenza and MERS. We're going into the first clinical trial in the middle of next year. And we'll also look at RSV included for 2020, but we won't go into clinical trials for that one. What we proposed in our grant was something they didn't ask us to do, but they liked the idea, and that was, we said, why don't you stress test us? So in our 3rd year, they will send us an email that has an unknown sequence, the equivalent of sequencing a new pathogen out in the field. We have to identify what virus group it belongs to, identify the protein that we need to express, and have a new vaccine ready within 16 weeks of getting that email. So that's our challenge in the 3rd year. So in a world of rapid travel and transportation, the impacts of demographic change and global climate change, there are lots of viruses that have the potential to spread and cause serious outbreaks. Of concern is that most of these are not detected until a full-blown epidemic, and this has been the case all the way along, of some unusual situation signalling the alarm. And it's usually too late to really affect effective control. We certainly didn't do that with SARS. But recent years, driven by WHO and organisations like CEPI, there's this realisation that we need to be epidemic prepared. And a significant coordinated effort with a lot of resources being poured into it is currently happening with multiple groups around the world to hopefully address any global health threat that emerges in the future by viruses, as I've mentioned right at the start, that emerge when, not if. And I'll just leave you with a large number of names of the people who are currently involved in our consortium that is delivering this particular vaccine. Thank you very much.
Fantastic talk, Paul. I'm going to give you a second to catch your breath. If you have questions, now's the time to write them down, wave them around in the air, and we will come around and collect them. There's already a couple come in on Twitter, so we'll get to those in a moment. While you're doing that, housekeeping, next month is our Summer Snake Spectacular. Try saying that 3 times fast. We have Dr. Brian Fry coming back. He's a few years and he spoke to us, and he's going to answer all your questions about snakes coming out this summer, as well as updating you on some of his latest research around venom and its application to medicine. So make sure you get along to that. A talk not to be missed.
That's 2 in one night.
Thank you. So, questions. I'm going to go to Twitter first of all, and Dom, bring me that first stack. So, first question is from Nicola, who asks, how the sperm penetrates and releases its genetic code into an egg reminds me of how viruses insert their genetic code into a cell. Is there any connection?
No.
Fantastic.
But it's a nice analogy.
Next question is, have human diseases spilled over into animal populations?
They have. There's plenty of evidence of the spillover events both ways. One of the reasons why we don't, or it's not encouraged that you engage physically with mammal species in the marine parks is the potential of actually transmitting, most particularly, respiratory pathogens. Similar respiratory viruses that we carry can actually infect those animals. So we have seen that before. We've also seen measles being spread into primate populations in close proximity to humans, and to devastating effect. So yes, you can see it both ways. And I just should point out, my colleagues at Science couldn't find the appropriate, the virus equivalent banner, so we brought along a lizard as well. So I was just thinking
Nearly
before,
as scary.
it's scary, but also it's quite possible that a virus could emerge out of a lizard, remote, but it's always possible that there's a spillover event from a lizard. So that's the connection, if you're wondering.
We've got a question here, jumping around a bit. Is it true that the smallpox virus is truly eradicated?
It is true, except for a couple of freezers. And in the virology community, there's been a long debate about whether that should be destroyed as well. So there's a few freezers in the former USSR, and there's some freezers in the US that we know about that still harbour smallpox. The argument for maintaining smallpox is that it has taught, as a virus, it has evolved quite unique strategies to suppress our immune response so that it could gain a hold. So we haven't fully understood that yet. So if we destroyed it, we would not give ourselves the opportunity to study that, to identify perhaps targets that we could identify for developing drugs, for example, to prevent other sorts of viruses doing similar things. So it's truly eradicated. Smallpox was a stupid virus. It only had one host, it was us. Once we'd eradicated it through effective vaccination programs and herd immunity, it had nowhere else to go. So that's one of the reasons why it was effectively eradicated.
Question from Twitter from David. Is it, but perhaps a little related, is it possible to develop a vaccine by reverse engineering immune cells to get an idea of what proteins matter?
Absolutely. It's possibly not exactly that way, but what we do is look. I mean, one of the things we do in our lab, and most virology labs do, most virology labs are also cell biology labs because viruses grow in cells. And so we are always looking at the very close interaction that viruses have with cells, and in particular, all of the proteins in a host cell that it requires for its own replication. So by studying that, identifying what host cells are important, we can also identify targets that we could design, not so much for vaccines, but more for antiviral therapies. Antiviral therapies up to date have mostly been targeted against virus-specific activities, but we've now seen that if we can target some host-specific activities that are not deleterious to the host, we can actually effectively break that virus infection cycle as well. So absolutely.
Okay, nice easy one. What is the worst case scenario for disease X?
Have you watched Contagion?
I played Pandemic.
Actually, Contagion, of all of the movies that have been made with this disaster scenario, the pandemic scenario, that's probably the one that's closest to virology real life. And the reason is they actually took on board a number of virologist consultants into the movie. And so couple of the issues that made for a virus that was able to disseminate across the world so rapidly was a high R0 value. I don't know if you remember the movie and remember them raising the R0 value. R0 simply refers to the capacity of one individual to transmit to another individual. So R0 of 1 is 1 to 1. And in that case, it would peter out very quickly. R to 2 means you would start to get an amplification event. Anything above that, and I can't remember what Contagion was, but it was a high R0 value, which meant it spread through the human population very quickly. So a highly pathogenic respiratory-borne virus is something we've always been fearing.
And perhaps a follow-up question from Howard. Does the community's mistrust in fact-based science and in health interventions such as vaccination make planning for pandemics more difficult?
Yes. It is challenging. I mean, it's frustrating, but I think it's our job to continue to explain and hope that facts matter in the end. Unfortunately, these days, facts don't always matter. Many people seem to be in a belief system where the facts won't drive through that belief system. I think it's a bit like herd immunity and vaccination. We just need to vaccinate enough people with knowledge and facts that will provide the protection to all. I mean, if there are people out there who don't understand the concept of herd immunity, it's why when we vaccinate with for some particular pathogens, it depends on the different pathogen. If we vaccinate anywhere between 85 to say 95% of the population, we can eradicate a disease in the population. Because at those sort of levels, two susceptibles just won't find each other. So you can have some people within a community who have those views and still be protected, and they will argue, I've never been vaccinated and I haven't had these diseases. Well, it's because everyone else is being vaccinated that's protecting the community from that spread. So maybe we can do the same with ideas.
Well, it's a good thing you all came along for your booster shot tonight of information. Go out and try and spread this particular knowledge disease with your friends. On that note, could you please put your hands together and thank our fantastic speaker this evening, Professor Paul Young. And we'll look forward to seeing you all back next month. Please join us for a drink outside. We'll see you soon. Thank you very much.
Thank you all.